Ladies and gentlemen, the distinguished lecture by Professor Randy Schekman now begins. Today, we are greatly honored to have as our guest Professor Randy Schekman to deliver a lecture at the university. First, I would like to invite Schekman Lee Professor of Life Sciences to Chinese University of Hong Kong, Professor Jiang Liwen, to deliver a welcoming address to introduce Professor Schekman. Professor Jiang, please.
Professor Randy Schekman, Professor Jimmy Yu, Professor K.B. Wang, Professor Feng Wenping, dear colleagues, students. So it's a great honor for me to introduce Professor Randy Schekman. So Professor Randy Schekman is one of the founding fathers of modern cell biology. He's an investigator of the Howard Hughes Medical Institute and also professor of the molecular and cell biology at UC Berkeley. So he has received many prestigious awards, okay, just to name a few, okay. He has been the member of the National Academies of Sciences since 1992, and he received the Albert Lasker Award for Basic Medical Research in the year 2002. Of course, in the year 2013, he received the Nobel Prize in Physiology and Medicine. He's currently the editor-in-chief of eLife. And Professor Randy Schekman has also been a very strong supporter for CUHK, also Hong Kong, in teaching, research, and service. So these are the three categories I think we have been evaluated by the university, in case you don't know. Okay. And Professor Randy Schekman is the chairman of the selection committee for Shaw Prize in Life Science and Medicine. And also he was one of the first teachers for our culture summer course. And also, he's a very strong supporter for our AOE research. To give you the example, so this was the culture summer course that was held in CUHK in August 2013. So Professor Randy Schekman was one of the speakers or teachers for that course, okay? And then in the same year, of course, he received the Nobel Prize. And this also showing you the supporting letter Professor Randy Schekman, also our VC, had provided in our AOE application. Okay. So just in case you don't know, 1 hour ago, Professor Randy Schekman just received the honor degree of Doctor of Science, okay, from the CUHK. So ladies and gentlemen, please join me to welcome the CUHK 2016 new graduate, Professor Randy Schekman.
This afternoon, I'm going to give a lecture of a very general nature about early and pioneering work of a number of people that really led to my career and which set the stage for our current understanding of how cells export proteins. To begin, I want to give a little background to my introduction to cell biology, which comes about through my very early interest in microbiology. So I'll discuss very briefly the origins and influences of the work in my lab over the past 40 years at the University of California, Berkeley. Well, some of you who were here an hour ago may have heard the story about this microscope. I'll be very brief. When I was in junior high school, I developed an interest in the microbial world because I had a toy microscope that allowed me to peer into the organisms that live in a drop of pond scum. I found this endlessly fascinating, and I resolved to buy a better microscope, and this is the microscope. It's a Bausch & Lomb student professional microscope that I bought at about the age of 12, and which was my constant companion throughout high school. During those years, I deepened my interest in cells and how cells work. And when I went to college at UCLA, I developed an interest in bacterial viruses and how they replicate their chromosomes. I learned the techniques of molecular biology as they applied to the mechanism of chromosome duplication. using small, circular, single-stranded DNA viruses, for example, PHYX174. During the years of my undergraduate studies, I became aware of the enormously influential work of the best DNA enzymologist of his generation, Arthur Kornberg. In 1967, Kornberg, in collaboration with Robert Sinsheimer at Caltech, did two beautiful studies that were published in sequential issues of the Proceedings of the National Academy of Sciences. Kornberg had, years earlier, purified an enzyme called DNA polymerase that copies the DNA template strand. It was the first enzyme to be described that took its instructions from another molecule, from a DNA template. And he reasoned through precise biochemistry that this enzyme was likely to be the enzyme involved in chromosome duplication. And the singular achievement described in these two papers was their ability to take DNA from the virus PHYX174 and to use Kornberg's enzyme to copy that DNA molecule to make an exact complement that was infectious, which means that the DNA polymerase followed its instructions faithfully in making a biologically functional molecule. So this and other arguments persuaded Kornberg and his colleagues that this enzyme was involved in replication. But 2 years later, in an explosive discovery made at Cold Spring Harbor, a bacterial geneticist by the name of John Cairns, who was deeply skeptical of Kornberg's conclusion, found in a random search of E. coli mutants, he found a mutant that was defective in the DNA polymerase enzyme, and yet these bacteria grew perfectly normally. How could that be? How could a bacterium grow perfectly normally, copying its chromosome without the essential enzyme for replication? Cairns showed that this mutant was sensitive to ultraviolet light, and he suggested that the polymerase was not the replication enzyme, but was rather an enzyme involved in DNA repair. And that, in fact, is what we now know to be the function of this enzyme. Well, these two papers, published while I was still a university student, were deeply influential on my own career aspirations because it taught me, on the one hand, the power of biochemistry to understand molecular mechanism, but I realized that that had to be paired with some physiologic function. In other words, combining biochemistry and genetics really was the most powerful way of proceeding to understand cellular processes. So I pursued my graduate career in Kornberg's lab, learned from the master how to take a complex problem apart to its component pieces, but then I decided that I wanted to explore a new area, one where there were no molecular interpretations that had been described, and that is how cells package proteins for export. I was greatly influenced in that direction by the work of the pioneering cell biologist of the early half of the 20th century, a man by the name of George Palade. Palade was himself a good friend of Kornberg's. I met Palade when he visited Stanford for lectures, but most importantly, I recall a lecture that he delivered immediately after he received the Nobel Prize in 1974. I had just moved from Stanford to UC San Diego, where I was a postdoctoral fellow, and within weeks of my arriving in San Diego, Palade flew from Stockholm down to the meeting of the American Society for Cell Biology, where he described his analysis of the complex interactions among membranes in a eukaryotic cell involved in the packaging and export of molecules like insulin. Here, for instance, is a thin slice through a cell that is professionally engaged in the production of insulin. It's the beta cell of the pancreas. And you see, I'm sure many of you have seen this before, you see a cell that is filled with organelles representing intracellular membranes that encapsulate insulin, in this case, in the form of these densely staining granules, and deliver it from the cytoplasm to the cell exterior by a process called membrane fusion. Now, as you'll see in the next several minutes, Palade used his skills in microscopy and also in cell fractionation to understand exactly where these granules, these vesicles, came from and how, at least at the level of the microscope, they delivered their content by fusion at the cell surface. All of the membranes in a cell, including the membrane that surrounds the cell, is composed of this unit structure. It's a bilayer consisting of two leaflets of molecules called phospholipids. Phospholipids display polar water-loving head groups, both facing the cytoplasm and the cell exterior, and they consist of an inner core of fatty acids that creates a hydrophobic barrier through which small molecules, unless they are desired, cannot pass either into the cell or out of the cell. That selective transport is achieved by these green structures. These are membrane proteins. They often span across the bilayer of the biological membrane, displaying surfaces on the outside of the cell or the inside of the cell. Some of these proteins may have specific channels through which small molecules pass into or out of the cell. Others may present a site on the cell surface to which hormones bind. These may be molecules involved in signaling, for instance, the receptor that recognizes and engages insulin and activates cells to take up glucose after you have a meal. All membranes in all organisms have this characteristic structure. Well, here is Dr. Pilati, later in his career, then a professor of cell biology at the University of California, San Diego, sitting behind his favorite instrument, the electron microscope. He did not invent the electron microscope, but he mastered techniques that allow one to preserve tissue specimens and to evaluate in thin slices through cells the internal architecture of cells. He was so powerful in his ability that he was the first in the 1950s to visualize the ribosome. That's the little machine in the cytoplasm that stitches amino acids next to each other to make polypeptide chains. He was also the first, as you'll see, to understand the function of a variety of membranes inside the cell that communicate with each other in the process of protein export. Here, for example, is one membrane that he characterized in some detail. It's called the endoplasmic reticulum, or ER for short. In cells of the pancreas that are professionally engaged in the production of proteins that are going to be secreted, these proteins originate on ribosomes, these dark structures, bound on the cytoplasmic face of this network of ER membranes. You can imagine the network of ER membranes as a kind of an interlocking connection of canals within the cell that sequester molecules that have been made by the ribosome and threaded across the membrane into the clear space. This clear space is physically connected in a canal network within the cell, and it collects all of the molecules that have recently been discharged from the ribosome. Eventually, most of these molecules will find their way out through this canal network out to the cell exterior. He not only visualized this structure, but he isolated it. He broke cells open. He purified the structure. He showed that these membranes, with the ribosomes bound to them, could actually continue to produce proteins and thread them across the membrane. Another membrane that he characterized was one that was actually discovered in the 19th century. This is a membrane called the Golgi apparatus. It was first described by an Italian cytologist by the name of Camillo Golgi, who in his career perfected various techniques of staining that would highlight different intracellular membranes. And by one combination of chemicals, he was able to highlight this structure as a series of membranes in a kind of a stack. Now, for 60 years after Golgi's discovery, the organelle was obscure. Many people thought it was some kind of a fixation artifact. It had no obvious function, yet it was found in almost all cells of animals and of humans. So it probably had some role, but it was really Pilati who figured out what that role is, and I'll tell you in a moment what he did to make that case. Well, at the end, after the Golgi apparatus, molecules like insulin become encapsulated within these granules, each granule surrounded by a bilayer membrane. At a certain moment, when the cell wants to discharge insulin to the cell exterior, these granules are pushed up so that they touch the inner or cytoplasmic surface of the plasma membrane, And the membranes, the two membranes then become so close to one another that they physically merge. The lipids that constitute these bilayers intermingle in a process called fusion, such that the membrane surrounding the granule becomes physically continuous with the plasma membrane of the cell. And when that happens, the content inside the granule is discharged to the cell exterior, where this material dissolves and insulin is eventually distributed into the bloodstream. Importantly, this fusion happens without breaching the permeability barrier of the bilayer. So the cell remains intact, everything inside the cell stays there, but only those things that are desired to be secreted are exported. Now, this particular event is actually repeated at many stations within the cell, and in many cells, even those that are not actually secreting proteins like insulin. For instance, as you're sitting there, this kind of cell is working very actively, although I have to tell you that this is a frog neuromuscular junction, so probably we don't have too many frogs in the audience. But your brains, I have to say, look very much like this, particularly those nerve cells that are in physical contact with muscle cells. At the nerve terminal, the same kinds of little vesicles gather, bind to the cytoplasmic face, and at the right moment, they discharge their content by fusion, their content being chemical neurotransmitters, molecules like dopamine that affects movement, or serotonin that controls mood, or acetylcholine, which is massively destroyed when patients succumb to Alzheimer's disease. So these chemical neurotransmitters, packaged at a high concentration within these vesicles, are delivered to the presynaptic membrane, where at a key moment of stimulation, the vesicle fuses and discharges neurotransmitter into the region between these two cells. The neurotransmitter diffuses, binds to a receptor on the muscle cell, and causes that muscle cell to contract. The same thing happens between nerve cells. There are trillion contacts between nerve terminals in our brain, always communicating with each other by this form of chemical communication. Now, this process can be visualized in a rapid sequence of events in a technique that was perfected by this fellow, John Heuser. Next slide, you see the resting nerve terminal of the frog neuromuscular junction. If these cells are stimulated by a brief electrical discharge, the vesicles that are at the nerve terminal rapidly fuse, discharge their content, and within 5 milliseconds in this time sequence, Heuser was able to see some of these nascent events where the vesicle has merged with the cell surface, and although you can't see it, you can well imagine that chemical neurotransmitters are diffusing into this cleft between the two cells. Well, Pallotti put all this together with some brilliant experiments that allowed him and then us to appreciate the stages in this process, and that is summarized here. He showed that newly synthesized proteins, like insulin, originate on ribosomes bound to the endoplasmic reticulum. They are then packaged into little vesicles that deliver their content to this structure, the Golgi apparatus. He was the first to show that this station is a temporal intermediate in the movement of molecules within the cell. He showed that this structure sorts and chemically modifies proteins, some of which are then packaged into granules, delivered to the cell surface, and then content discharged by membrane fusion. Now, he won the Nobel Prize, quite rightly so, in 1974 for his pioneering discoveries. But when he presented this work at the meeting of the American Society for Cell Biology, I was struck by the absence of molecular information about this pathway. This is a complicated pathway. It's quite obvious when you look at it that there must be many different genes and proteins orchestrating this process. And yet in 1974, not a single molecule associated with this process was known in any depth whatsoever. There were no genes, no proteins that were known to organize this process. And so I decided in my own independent career to develop a way of studying this, not in the typical tissue or organisms that Pallotti and the pioneering cell biologists used, which were largely mammalian organisms, but rather in a simple organism that has a nucleus. I moved in 1976 to UC Berkeley, where I began to do my studies on baker's yeast. Now, I was motivated by this choice by the pioneering work of a yeast geneticist by the name of Leland Hartwell, who, with his colleagues at the University of Washington, had used traditional, really classical genetics to dissect the sequence of events that a cell relies on to progress through the cell division cycle. He did this by assuming that there were genes required for cell division that, when mutated, might produce forms that were crippled at a particular stage in the cycle of events that leads to a dividing cell. So he isolated a series of mutants, calling them cell division cycle or CDC mutants. And in these mutants, when the cell is arrested because of the absence of a particular gene product, the cells accumulate with a terminal morphology, a terminal appearance that it reflects an arrest at a particular stage in the cell division cycle. So you see many different phenotypes associated with mutants. And he and others were then able to characterize these genes, and now we know a great deal at a molecular level about the logic and organization of the cell cycle, largely through Hartwell's initial work. When I had a look myself as a postdoctoral fellow at a culture of yeast cells, there were certain things about the cell that looked interesting, at least in respect to the process of protein secretion. Here's a cluster of yeast cells seen in an electron microscope called a scanning electron microscope. You see the kind of surface features of a yeast cell. A yeast cell grows not by division in half, but rather by a budding process. So a cell, early in the cell division cycle, picks a part of the cell surface to expand, and during the 90 minutes or so of cell division, that bud, the new daughter cell, grows in preference to the mother portion of the cell. And finally, after 90 minutes, when the nutrient conditions are right, the cell splits in half, and the daughter and mother cell can proceed to produce new buds and grow indefinitely. Now, if a simple inspection of this would convince anyone that new surface material, molecules that comprise this surface, must be added to the bud envelope, to the bud membrane. In fact, if you look inside the cell by cutting a thin slice through a yeast cell, you can see evidence of that. Here is a thin section through a yeast cell. It looks not nearly as dramatic as a pancreatic beta cell, but there are some elements, some membranes in yeast cells that reflect the normal process of protein export. For instance, there's a structure called the vacuole, which is the yeast equivalent of the lysosome. There are membranes of the endoplasmic reticulum that adhere to the inner surface of the plasma membrane. But the most important thing to me, at least on initial inspection, was the appearance of a cluster of small vesicles under the bud portion of a growing yeast cell. These look very much like the vesicles that are in a nerve terminal or vesicles that are about to fuse at the membrane. And we imagined right at the outset that these vesicles would carry proteins for export into the cell wall region that surrounds a yeast cell, but even more importantly, that the membrane of this vesicle could be the building block for the assembly of the plasma membrane of the cell surface. So if you have a bud and you start inserting new membranes in the form of vesicles, the cell would enlarge step by step. And if that's true, then the genes required for the production, migration, and fusion of those vesicles would, like Hartwell's cell cycle genes, be essential for cell viability. Now, Hartwell relied on techniques that microbiologists have used for years, and we too relied on the observation that essential genes can be crippled partially by the introduction of mutations that change a key amino acid residue in the protein, creating a protein that is thermally unstable. Yeast cells grow at a range of temperatures, from room temperature up to human body temperature. They grow perfectly normally. That means that all of the essential proteins in a yeast cell can perform their function anywhere in this range of temperature. However, sometimes a mutation introduces a changed amino acid into the folded structure of a protein such that that protein no longer functions properly when the organism is raised to human body temperature. And if that happens, the cell will die. Sometimes those mutations can perform that function if the cells are maintained at room temperature. And if that's so, then one has a convenient way of discovering a gene that is involved, that is essential for growth, by growing the cells at room temperature under conditions where this protein functions, and then warming those cells to human body temperature, you can explore what happens as the cell dies. So those are essential genes identified by temperature-sensitive lethal mutations. Now, much of the work, the early work in my lab was conducted by a brilliant graduate student by the name of Peter Novick, shown here in a picture taken in around 1978. Note the long hair. I too had long hair back then, but at least I have more hair than Peter does now. But Peter has gone on to his own brilliant career. In fact, he is the George Palade Chair of Cell Biology at UC San Diego. Well, we began to explore conditions that would allow us to identify temperature-sensitive mutations in genes required for protein secretion. But this was all done in the context of the University of California, Berkeley, the birthplace of the student protest movement. And anything that you do of any controversial nature at Berkeley engenders criticism and protest. And so since we were proposing to kill living organisms, we too suffered from protest movements. End the torture in the labs. Yeast have feelings too. We were able to overcome the objections. The Animal Experiment Subject Committee recognized that yeast are not sentient beings, and we have since killed trillions of yeast cells and have not heard a scream from any of them. Well, in May of 1978, Peter had obtained the first mutation that seemed to block protein secretion. These cells were temperature sensitive for growth, and when the culture was warmed to human body temperature, 37 degrees, enzymes that would normally be secreted outside of the cell instead built up inside the cell. At that point, just by chance, Dr. Palade visited UC Berkeley for 2 special lectures. I had the pleasure of introducing him to my new work, and more importantly, Peter organized a dinner hosted by the graduate students in honor of Dr. Palade, and Peter told Palade about his work, and Palade said, well, of course, you must now cut thin sections through this mutant to see what it looks like. Shortly thereafter, one of the great moments in my scientific career, Peter excitedly called me from my office on the 6th floor of our building down to the basement where our electron microscope was housed to show me one of many of the following image. This is the first mutant. We knew that it accumulated secretory proteins inside the cell, but we had never seen anything like this, a cell that instead of having the small number of vesicles congregating under the bud portion, now fills its entire cytoplasmic volume with thousands of vesicles that continue to be produced, but which fail to engage the plasma membrane and then and thus allow the cell to expand. These cells eventually die of a kind of molecular constipation. We know this gene, which we call SEC1, is found in the genomes of all organisms that have a nucleus. It's certainly found in the human genome. And we know that that protein, the SEC1 protein, is organized wherever a vesicle must fuse with the target membrane, even at the synapse, at the presynaptic membrane. That protein helps to organize the fusion of a synaptic vesicle. This process of protein secretion is evolutionarily conserved over 1 billion years, having first evolved in microorganisms. The parts that organize this pathway have been handed down over the eons to our current legacy in the human genome. Now, Novick, excited by this discovery, decided that it would be important to develop an approach to enrich for more mutants. Rather than just looking at random mutants, he decided to try to find a way to enrich for mutants. And he recognized in looking at this mutant under the microscope that when the cells are warmed to 37 degrees, the cells fail to enlarge, but they're continuing to make everything. They're making all these macromolecules. So he reasoned that the cell must increase its internal density, grams per cc. Its buoyant density must increase. And that gave him an important insight, because it seemed possible then that we could select, or at least enrich, for more mutants by separating mutant cells from normal wild-type cells on a density gradient designed to distinguish dense mutant cells from light wild-type cells. So he then did another incredibly simple experiment that I still remember in my mind's eye, that he lined up on his bench. He took a tiny proportion of SEC1 mutant cells, suitably marked so that he could distinguish these cells when they formed a colony on a Petri plate, and he mixed those cells with a huge excess of wild-type cells. And he took the mixture, he incubated that at 37 degrees, and then applied the mixed sample, to the top of a centrifuge tube containing a medium that allows cells to be resolved on the basis of their buoyant density. After just a few minutes spin, he took the tube out, he punctured a hole in the bottom, and he collected drops representing 10 fractions, 11 fractions from this gradient. And when he displayed colonies on these 11 plates across his desk, his bench, we could see that all of the mutant cells were in the bottommost 1 fraction, whereas all of the wild-type cells remained at the top of the gradient. So this is a biochemist's idea of a genetic selection, using a density gradient to purify mutants, and he did so then over the next year and a half, and obtained an additional 220 independent mutations that affected many different genes. Our original collection from this genetic analysis defined 23 different genes that act at different stages along the secretory pathway. There were 10 genes that produced mutants that were just like SEC1, that accumulated lots of vesicles. That means there are at least 10 different proteins that operate at that last step in the pathway. A smaller number of genes, 2 to be precise, produced a very different structure shown here. This is a yeast equivalent of a Golgi apparatus. It's a structure that is rarely, if ever, seen in a yeast cell, because yeast cells never are normally arrested at this stage. But when you arrest traffic at that stage, the cell continues to make this membrane and to build it up, looking very much like an animal or a plant cell Golgi apparatus. These cells, like others blocked in secretion, when returned to room temperature, restore the pathway and deliver the accumulated material to the next station. Another different phenotype is seen here. 9 genes produce mutations that block at an earlier stage in the pathway, causing the endoplasmic reticulum to accumulate to a larger extent than is normally seen. It is distorted in its width. The nuclear envelope, which is physically connected to the ER, is also distorted. And here again, when cells are returned to the permissive temperature, the material that had built up is discharged and moves along to the next part of the pathway. Now, at this point, we published several papers, and we're very excited to get into molecular details, but we were a little surprised, and in a way disappointed, that of all of the mutants and all the genes that Novick isolated, not a single one seemed to affect the very first step in the pathway. We and others predicted that there had to be a channel in the ER membrane through which secretory proteins that are being made by the ribosome pass on their way into the canal network of the ER. And yet we didn't find any mutations that seemed to block that step. So several years later, another really brilliant graduate student by the name of Ray Deshay joined the group and devised a quite clever genetic selection that allowed us to define genes that constitute that channel in the ER membrane. Let me show you Ray first. Here's Ray at a banquet celebrating other discoveries in the lab. There he is. Ray has gone on to his own stellar career as a professor at Caltech. He was just this past month elected to the U.S. National Academy of Sciences. This is his wife, Linda Silvera, who was also a graduate student at the time, and another graduate student, Linda Hickey, whose work I'll describe in a few minutes. So here's Ray's experiment, really very clever genetic selection. Ray relied on the fact that most enzymes in the cell that act on substrates do so in the cytoplasm. And it is unusual, it would be unexpected for the substrates for an enzyme, charged substrates, to be able to pass back and forth across membranes, for instance, of the endoplasmic reticulum. And yet we also knew from other experiments that if you take an enzyme, like the enzyme that catalyzes the last step in histidine biogenesis, converting histidinol to histidine, if you take that enzyme and you take the gene for that enzyme and you stitch at the beginning of that gene a piece of sequence that encodes the signal that proteins need to penetrate across the ER membrane, such an enzyme signal hybrid can be artificially induced to be translocated into the lumen of the ER, where this enzyme will not have direct contact with its substrate. So you can force the cell to move this away to a location where it can't create new histidine for cell growth. So this is the goal of a geneticist, to create a situation that allows you to select for mutants. This cell will not grow on histidinol as a source of histidine. So if you expose this cell to a chemical that causes mutations, occasionally, by random hit, a mutation will be introduced into the gene that encodes, for example, the channel in the ER membrane. And if that mutation partially cripples that protein, you may produce a cell that leaves behind enough of the histidinol dehydrogenase signal-containing protein to catalyze the conversion of histidinol to histidine. So that sounds like a lot, but all it involves is exposing the cell to a mutagen and then plating it out on a simple medium into which histidinol is provided in place of histidine, and you look for mutants that grow because they have left this protein behind in the cytoplasm. Well, that procedure resulted in the selection of about 6 new genes that we had not previously discovered, the first one of which, just by chance, Sec61, is the gene that encodes the channel in the ER membrane. And we now know that that channel is the same in the human genome and the same in bacterial and archaeal membranes involved in secretion by prokaryotes. Well, let me put these all together in the form of a map, a kind of a contour of the secretory pathway in yeast. This looks very much like what Palade described by his pioneering work, but importantly, at each stage we can populate an element with a set of genes predicting the existence of proteins whose function is required to move things together from one station to the next. Now, in the mid-1980s, when this map was first established, it became possible to clone these genes by genetic complementation, to obtain the wild-type copy of the gene, and then to sequence that gene to deduce its nucleotide sequence and thus its amino acid sequence, and thus to predict at least the chemical nature of the proteins that are encoded by these genes. It was even possible then to show that the same genes exist in the genomes of higher organisms, even the human genome. And yet, for most of these genes, the mere information of the amino acid sequence of the gene did not tell us anything about what the protein does. You can't necessarily look at a gene and its protein sequence and immediately figure out what chemistry is catalyzed or what exact process the molecule participates in. And it was for this reason, indeed, it was my conviction all along that one would need a cell-free biochemical reaction that reproduced in the test tube some element of this pathway with isolated membranes and soluble proteins, that one would need that kind of Kornberg style of biochemical reconstitution ultimately to characterize the proteins and to understand what chemistry they catalyze. So we resolved over a period of years to devise a cell-free system that would reproduce this, and I had the great good fortune in the mid-1980s to have another brilliant graduate student, David Baker, who came to the lab and within weeks had devised a biochemical reaction that reproduced in the test tube the first half of the secretory pathway. Now I want to take a step back to tell you a little bit about the origin of this idea that I'm going to share with you about how we proceeded to use this biochemical reaction. It begins many years ago in the laboratory of Bill Wood and Bob Edgar at Caltech. Edgar was a bacterial virus geneticist. He had used mutations in genes of the bacterial virus T4 to deduce the events involved in the production of an infectious virus particle. But these were experiments conducted in the early 1960s, long before the advent of DNA cloning, long before it was possible to sequence genes and discover the nature of mutation. And yet, he had genes that he knew were involved in bacterial virus assembly. In 1965, a young assistant professor, freshly minted PhD student from Paul Berg's laboratory at Stanford, a man by the name of Bill Wood, moved to Caltech, and having been trained as a biochemist, he joined forces with Bob Edgar to do what I consider to be one of the classic experiments in the history of molecular biology. They devised what I call a biochemical complementation assay to understand the nature of the genes that had been mutated in Edgar's genetic studies. Here's the experiment. It is brilliant and simple in concept, my favorite kind of experiment. What they did was they took two different cultures of bacteria, and each one was infected with a mutant virus, which by itself was incapable of producing infectious virions. Now they knew, because of Edgar's genetic studies, that if these bacteria were infected by the two viruses into the same cell, genetic complementation would allow the bacterium to produce viruses and to rupture and to make many more virus particles. But if the bacteria were infected separately, nothing would happen. Then they reasoned, if this process could be reproduced in a broken cell preparation, perhaps virus particles would form if the lysates were produced of these two infected bacteria and mixed together in a test tube. Perhaps the defective gene product in this test tube would be complemented biochemically by the normal counterpart in this test tube, and vice versa. And indeed, the data were quite dramatic. At the beginning of the incubation, virtually no virus particles are seen, but in the course of a 30 or 40-minute incubation, more than 1,000-fold increase in virus particles are produced in this biochemical reaction. So this, to me, was a lesson to be learned. It was a lesson that I learned and applied as a graduate student in Kornberg's lab, and it was a lesson that I repeated in my own work when Baker joined the lab and developed the cell-free system that I described a moment ago. Now here's the experiment, the ultimate experiment that is our version of the Wood and Edgar experiment. It was an experiment conducted by Linda Hickey, whose picture I showed you a few minutes ago. It's another very simple experiment. Linda produced a cell, a mutant cell, broke it open, a mutant that's defective in a particular gene product. She broke the cells open to obtain cytosol, soluble proteins from this mutant cell, and she obtained membranes from a wild-type cell. She also produced a cell, a mutant cell, into which she introduced a wild-type copy of this gene on a plasmid, such that cytosol extracted from this transformant would be expected to have at least some of the wild-type gene product. She mixed these two different protein fractions with wild-type membranes and incubated them in the cell-free system that Baker had devised and measured the extent of transport at different temperatures. At the lowest temperature, 15 degrees centigrade, both cytosol fractions stimulated transport well above a background of an incubation conducted without cytosolic proteins, that is, one that had only membranes. This reaction required cytosol as well as membranes. But importantly, as the temperature of the incubation increased, the mixture that contained only mutant cytosol was progressively reduced in its ability to sustain transport, whereas transport was observed nearly normally, even at the higher temperature, when the cytosol carried at least some of the wild-type copy of this protein. So this was, to me, was one of the great experiments in my lab, because it immediately gave us the opportunity to fractionate this system and to use the Wood and Edgar logic of biochemical complementation to purify individual normal gene products using this assay. Well, at the same time, Chris Kaiser, a postdoc in the lab, had used an elegant combination of genetics and localization and a thin section microscopy to further define the genes in yeast cells that are required for the formation of transport vesicles, that is, genes that participate in vesicle budding, as opposed to genes that are involved in their traffic to the Golgi apparatus. And across the bay, then at Stanford, Jim Rothman and his colleagues had purified 2 proteins in an assay, a biochemical reaction that measures traffic within the mammalian Golgi. They isolated 2 proteins. They cloned the mammalian genes that encode these proteins and discovered that the mammalian cell has the equivalent of these 2 genes in yeast cells, the Sec17 and 18 genes. And then it became clear at that moment that the 2 labs were working really on the same thing, one by a very different approach, a purely biochemical approach, and another on a genetic and biochemical approach. Well, the reaction that Baker had devised and that Hickey used to develop biochemical complementation, we now know to require probably several dozen different genes and proteins. Biochemically, it was clear that it was complex, and it seemed difficult at best for us to try to fractionate that system to obtain all of the proteins and to understand mechanism after all that fractionation. And so Michael Reksash joined the group and devised a very simple biochemical reaction that allowed us to focus on just the first step where a vesicle buds from the endoplasmic reticulum. And here's the principle of his assay. Again, a simple, elegant assay, the kind that I really favor. Reksash observed that in the mechanical lysate that Baker produced that sustained traffic, the membranes of the endoplasmic reticulum remain very large envelopes. They're so large that they will sediment at a very low speed when you put the sample in a centrifuge tube. However, if you incubate these large membrane envelopes with yeast cytosolic proteins and a source of energy in the form of ATP, small vesicles are produced that are so small that they cannot be centrifuged into a pellet at a moderate speed. Instead, these vesicles must be centrifuged to obtain a pellet only after very high speed. So this is a really trivial assay. It involves just a couple of different centrifugations of a sample. And this allowed us to quantify the production of vesicles that bud from the endoplasmic reticulum. And Reksash was able to show that the event reproduced in the test tube requires the same genes that I showed you a moment ago that Kaiser had found to be required for budding of vesicles in intact yeast cells. So using the biochemistry and molecular biology that was then available, we were able to purify all these proteins and discover exactly how they cooperate to bud a vesicle from the endoplasmic reticulum. Here is an example, after several years, of our achievement. This was the work of Charlie Barlow, a postdoctoral fellow, in collaboration with another wonderful morphologist by the name of Lelio Orci at the University of Geneva. Barlow found that the set of pure proteins that Linda Hickey and others had isolated that reproduced the formation of vesicles in the test tube, when mixed together in a cell-free reaction, produce vesicles that can be seen to carry a kind of a fuzzy coat that surrounds the entire surface of the vesicle. This was really quite a striking observation, and we discovered that the proteins that we require for the production of vesicles actually constitute subunits of this apparently fuzzy coat. These genes are also conserved evolutionarily, and they are found in the human genome and affect traffic in human cells. Now, we know through the work of other investigators who have used the techniques of X-ray crystallography and structural biology, we now know that this coat, far from being fuzzy, is actually very highly organized. There are 2 layers of the coat. There are proteins that touch the cytoplasmic surface of the ER membrane and grab membrane proteins that are designed to be pulled along the secretory pathway. And then there's a scaffold that forms a regular kind of geodesic dome or polyhedral network that envelops the membrane, forms the vesicle, and pinches it by fission from the membrane from which it originates to produce this coated vesicle intermediate. Now, we were content to study this for many years and continuing our studies both using genetics and biochemistry with yeast. But then it became clear that some of these genes have been mutated in human diseases. This was surprising to me because the genes that we discovered in yeast are single essential genes. A point mutation is enough to kill a yeast cell that has these single copies. But the genes that encode these proteins in humans are duplicated, at least, and that means that there's some functional redundancy. In fact, here they are. These proteins that operate to form the coat have, some of them have 2 copies, some of them have 4 copies, and as a result, mutations have arisen in the human genome that cripple one of the 2 copies and produce a tissue-specific pathology. One such tissue-specific pathology came to our attention through the work of a clinician at Johns Hopkins University, Simeon Boydjev, who reported to us a patient family, a Bedouin family from Saudi Arabia, who had a single mutation in an invariant, highly conserved amino acid residue of one of the 2 copies of the human SEC23 gene. These patients present with a craniofacial disorder, with bone fragility, and with a curious inability to complete the soft spot, the fontanelle on the top of their head. It's one of a very large number of craniofacial disorders, but this one was a very specific one as a result of a single mutation in this gene. We obtained a sample of skin cells from the patient, from his mother, a heterozygous carrier that shows no pathology, and compared the impression of the cytoplasm of these cells with that of a normal skin cell, a fibroblast from a normal cell. Here are the patients. They have brittle bones, facial dysmorphism, cataracts, and a failure in the closure of the fontanelle. Skin cells from the patient were compared in thin section electron microscopy to wild-type normal human cells or cells from one of the carriers. The ER membrane from a normal cell is fairly smooth, some granular staining material. From one of the heterozygous parents who showed no pathology, some apparent distortion of the ER, but not enough to cause any disease. But remarkably, the children, the homozygous children, at least one of them, has an enormously distorted endoplasmic reticulum. This was even more dramatic in its defect than the original mutant that we found in yeast cells. We now know that these cells, although they grow normally, nearly normally, are delayed in the secretion of proteins such as collagen, which build up in the interior of this membrane. Well, the work continues. We have many new investigations on other pathways in cells. Tomorrow at CUHK I'll give a summary of some more recent work on the problem of how collagen is secreted from cells. Well, let me put this all together in the form of several lessons that we learned over now almost exactly 40 years at Berkeley. Our initial genetic studies documented that secretion and the assembly of the plasma membrane surrounding a cell are physically and functionally linked. Palade was able to show that these organelles are temporally related, but our genetic studies further documented that these organelles are actually required as obligate intermediates. We were able to clone these genes, and some of the genes are so evolutionarily conserved that the same gene acts in bacteria, in archaeal species, in eukaryotic microorganisms, and in plants and humans. And that, for instance, is true of the gene that encodes the protein that forms a channel that completes that first step in the pathway. More recently, we've been able to discover this novel coat. We can show that it binds to and brings along cargo molecules that are designed to be exported to the cell surface. We also know that it's physically involved in pinching to form a membrane. And we can explain at least some diseases where the genes have been affected by the tissue selective expression of some of the paralogs of this pathway. All this work was well and good, but then my life changed a couple of years ago. On October 7th in 2013, I was awakened in the middle of the night, and then I came to the lab to discover that every member in my lab, for instance here, you can see Yusong Guo now has a yellow mustache. Really, I haven't seen that mustache on you, Yusong. They all showed up. Even my wife and my secretary had donned mustaches for the celebration that morning. Several weeks later, I received an email inquiry from the Nobel Museum in Stockholm. Every year, laureates are asked to provide some artifact from their past that reflects their interest in science. And it dawned on me in very short order that I had that old microscope, that sustained my interest in microbiology throughout high school. Of course, got put away in my parents' garage. They sent it up to me, and regrettably, neither of my children demonstrate any interest in science. It sat collecting dust in the wine room downstairs, and after that message from the museum, I went down, dusted it off, gave it to my lab manager, and we shipped it off to Stockholm. And if you happen to visit Stockholm, come by and see my old microscope here on display, together with a story about how I had to run away from home to the police station to extract this microscope from my parents. Thank you for your attention.
And for imparting your valuable knowledge on the subject. Please take the seat on the stage. Professor Schekman has kindly agreed to take a few questions from the audience. And I would like to invite Professor Zhang again to come forward to the stage and conduct question and answer section together with Professor Schekman. If you would like to ask any question, please raise your hand. Professor Zhang will indicate if your question is being taken, and our staff will pass the mic to you.
Hi, my name is Jonathan from CUHK. Okay, so I just want to ask a question, a very general one, about your scientific discovery, right? How do you know that you have finished and exhausted all the genes, that you know that you found enough? And how do you know that you should keep going?
That's a good question. We now know probably almost all of the genes, but that's many years later. So I think what I think your question is, how did I decide at a certain point that I had enough and it was time to move on to discover what the genes do? After that year and a half where Novik collected an additional 220 mutants and defined 23 different genes, I decided, since we didn't know what any of them did, that it was time to start exploring, devising a way that we could actually figure out what these genes do. The most obvious way was to clone the genes. It was then possible to do that. It was a little laborious, but it was possible to do, and some of the people in the lab and other labs started to do that. But I was certain, because of my background, that the genes that we clone wouldn't tell us much about what the proteins do. I described the logic of that. At the time, cloning and sequencing a gene, if the gene didn't have an equivalent gene of known function somewhere else, would tell you nothing about what the gene product did, other than that it had a predicted molecular weight and a certain sequence of amino acids. So at that point, which was now in the early 1980s, I decided that it would be essential to devise a biochemical approach, that is, of the sort that I told you about here that Wood-Edgar pioneered. But it turned out to be very difficult. Difficult for two reasons. One is that people who joined my lab at that time saw that the genetic approach was working very well, and they only wanted to make more mutants and clone genes. I tried unsuccessfully to persuade a series of postdoctoral fellows and graduate students to be bold and to go in and make an extract and try to reconstitute a reaction. And very few were willing to do that. Eventually, after a few years, this fellow, David Baker, knew himself how important it would be to achieve that. And in his case, within 2 weeks, he had devised a means of breaking the cells open and measuring traffic. So that's a long-winded answer, but it was a tactical decision on my part to say we had enough genes and now it was time to figure out what the genes did. Now, it turns out other laboratories then went on and isolated more mutants and more genes, and that was good. But I wanted to really understand what the genes did, and so we had to change our tactic.
Thank you very much for your nice talk. My name is Carol from CUHK. I want to ask a question that, do you think any different functions exist in the contact CUP2 vesicle and the pre-budding complex of CUP2 that without the outlayer coat CUP2?
That results in the uncoating of CUP2.
Yeah.
Yeah. Okay, that's a very specific technical question about issues that I didn't get into in this lecture. So please come tomorrow. I'll be more detailed tomorrow. This young woman obviously knows more than some of you know about the details of how the coat is assembled, but so let me be very brief. The coat begins to assemble as a bud forms, and then once the vesicle is completed by fission, the coat is shed. And as I'm sure you know, that is stimulated by the hydrolysis of GTP by one of the proteins that's intrinsic to the coat. So the crucial question, which still really can't be answered satisfactorily, is how does the hydrolysis of GTP know to be completed as the vesicle completes its fission, rather than to prematurely uncoat a bud before it has a chance to separate by division? Probably I would guess it involves proteins in the membrane, because as you may know, if you take these proteins that we've purified and you attempt to do this reaction on a synthetic membrane that has phospholipids but no membrane proteins, you can't complete a budding process because the coat prematurely discharges. And so my guess is that there are proteins in the membrane that retain the coat long enough until the bud completes its fission process. So what those proteins are, they could be cargo molecules to which the inner layer of the coat adheres, that keeps the inner layer of the coat present until the coat is separated. Or there could be some catalyst that's required for budding that we've missed so far. So there's certainly more that needs to be discovered. Is this a subject that you're working on? She smiles. Okay.
All right.
She's not going to reveal any secrets.
So I guess you're from David Benfield's lab, no? Okay. CUHK, okay. All right, sorry.
And hello, I'm Bill. I'm a normal person. And
You're a very normal person. Is that what you said? Okay.
And then
I'll have to take your word on that.
I am not very familiar with biology, and then I am going to ask a stupid question. And
those are the most dangerous ones.
in Hong Kong, there's lots of people, and we always have the problem of the traffic jam, that the car, they always get together, and we have and take lots of time to staying in the public transportation, and you're studying the cell traffic inside for 40 years. And do you have any insight or any that how the cell can solve this problem? We do not see any transport problem in the cell. Thank you.
Well, I guess what I can say is the cell is smarter than than your local transit authority. Yes, the solution is public transportation. You see, when molecules are secreted by cells, they don't go one by one in a carrier, they go in bunches. And that is much more economical for transport. So if everyone took public transportation and didn't take the individual cars, the traffic would be much less severe. And that's what we've learned from cells.
Hi, I'm Hoi Wei, a medical student here, and I don't know much about cell biology, so please forgive me. And I just want to ask are there like some master genes like that control the expression of those SEC genes? Because it seems that all the genes you've mentioned are like they're distinct. How do the cell knows that the previous process, such as the budding of the vesicle, has been completed? And and it can proceed to the next step. And how do the cell know that? Like, because there are so many different chemicals being produced in the cell, how do they like not get messed up? Because there's so many signals being involved and stuff going on.
Okay, you say you're not a biologist, really?
No, I'm just ordinary medical student. Like,
Okay, I can't answer every aspect of your question, but there's one that you hit on right at the outset that I can answer, and that is how does the cell orchestrate the production of the machinery that's involved in this process? It turns out that cells that are professionally producing secreted proteins, like in the pancreas, or cells of your immune system that become differentiated to produce antibody molecules. The genes of the pathway that I've described to you this afternoon are themselves under master regulators of expression. In fact, there is a very small number of specialized transcription factors. These are factors that in the nucleus promote the production of RNA molecules that encode the machinery that I described. And when an antibody-producing cell arises through development, those genes that produce these transcription factors are turned on, and they in turn turn on the machinery so that the cell can now turn its metabolism in the direction of organizing secretion. So at that level, there's actually some very interesting and very specific information that is known. That doesn't happen in yeast because yeast cells are always doing this. I mean, when they're growing, they're always wanting to produce this machinery. But in more complex cells, that organization is itself under control.
Thank you for your wonderful talk. My name is Alfred Chan in School of Biomedical Sciences. I have two questions. So during your lecture, I see some of the very brilliant experiments being performed to solve some questions. And whereas nowadays, lots of people do next generation sequencing. So everything is being sequenced and getting some data. But in contrast, your ways of doing experiments is very fascinating, and you think a lot. And I want to ask how you come up with the ideas of different ways of doing experiments.
Okay, well, that's a tough one. First of all, in any generation, it's always true that a lot of people swim on the tide of technology and apply the latest technology to generate a lot of information. Next generation sequencing. The same was true of other seemingly simpler techniques in my generation. But there still is, and always has been, a way of looking at a problem and trying to think of simple ways of solving the problem, rather than using the latest technological development to solve a problem. And maybe it's an old-fashioned way of thinking about science. But as I emphasized in my talk today, what really appeals to me are the simplest experiments, involving simple logic and simple tools. When we started our work, it was good enough to use toothpicks and Petri plates and agar and medium and simple chemicals. And I still think it's true today that people spend a lot of time thinking about the latest technology in trying to solve their problems. But sometimes simple is the more elegant solution. So I have no unique ability to do this. I think there are plenty of people who think that way. And it's just to convince yourself that it's possible to think that way and not to necessarily rely on technology to solve a problem.
Thank you. My next question is not academic. Very obviously, you in your lecture, you have a lot of great students and postdoc that help building your story and in your journey of science. So I want to ask, what is your tricks of working with students and postdoc?
What is my what?
Tricks.
Tricks.
Yeah, in a very productive way. Yeah. How to manage your group of scientists.
Yeah. Well, I don't know if I have tricks, but there are different styles of success. Okay, let me tell you about my PhD mentor, Arthur Kornberg, who was, by any measure, one of the great biochemists of the 20th century. His style was, he was very demanding. You had to do exactly as he said. He didn't brook any dissent, and he, in a way, operated by fear. And it worked. It worked. There were people in his lab who did the best work of their life when they were in his lab. But subsequently, many of them failed because they didn't have him dictating their work. So now I didn't try to embrace that style because it's not in my personality to do that. But it works. That's a style that works, and some people are like that. I try to encourage people by being enthusiastic. I get excited about some project, and I try to, share my enthusiasm. And if students can be motivated by that, then that kind of a student will work well in my lab. If the student needs direction, day-to-day direction, they're probably not going to work well in my lab. So I look, when I'm taking new people in the lab, I look for people who I think can be more independent and can plan their own daily tasks, and then can motivate themselves by being enthusiastic about the project. And that works for some of my students, but not for all of them. And so I think inevitably, with that style, there are going to be some people who don't succeed. But my feeling is that the Kornberg style may work while you're in that lab, but you're not creating a great scientist by that approach. If someone has the enthusiasm, then of course the lessons that you learn from someone like that can be really powerful. But if they don't have the fire, any amount of coercion is not going to turn someone into a great scientist. So I don't know if that's a trick, but it's a style. Just to leave you, there are different styles, and there are different styles that can succeed. And so I think when a young person is starting his or her own lab, they have to sort of figure out what their own style is and try to optimize that for progress.
And last technical question is, what do the Nobel Prize mean to you? And how has that prize changed your life, both academic and non-academic? Yeah. Thank you.
Well, of course, it's a great honor. And it means a lot personally. It has changed my life. I go around giving lectures like this more often than I used to. But it has also provided me with an opportunity to express my views on subjects that are not necessarily directly related to my science, because when you win the Nobel Prize, you have a kind of a soapbox that you can stand on. And some people who win the Nobel Prize retreat back to their laboratory and continue to do what they're good at, which is fine. Some people go permanently on the lecture circuit and never get anything else done. And then what I would prefer to do is to use my whatever influence I have to try to change things in certain ways. And there are three things that I feel most strongly about that I've expressed and acted on. One is, you may not have been there a couple hours ago, but I feel, when I spoke to the convocation, I feel very strongly about public higher education, which I see under attack, particularly in the United States, with a loss of support at the state level for higher education. And so I have done whatever I can to help my institution raise money to replace the money that's not being provided by the state. I also feel very strongly about the importance of basic science, as opposed to what now is the buzzword, translational science. I think that we are losing sight of the importance of basic discovery as the foundation for application. And I think governments and university administrators everywhere need to appreciate the importance of basic science as the foundation for technological advance. In the United States, for instance, there's increasing pressure on scientists to do applied work in their own laboratory. for biomedical scientists like me to be turned into drug discovery laboratories, which I am personally ill-equipped to do, and which I think most of my colleagues are not particularly adept at. I feel that the basic discoveries should be made in universities like this, and that information should be available to the private sector for its application. So whenever I have a microphone like this, I say things like that. And finally, I've taken a more direct role in an issue relating to how we evaluate scholarship. And that is specifically how publications are evaluated and the value associated with publication in certain journals. Some of you may know my views on this subject. I've taken a very strong stand against what I refer to as the luxury journals, and these are specifically Nature, Cell, and Science, journals that promote themselves as exclusive venues for so-called high-impact work, but because of their exclusivity and the strong feeling that scholars have that they have to have their work in these journals, mistakes are made. The work is hyped beyond recognition, shortcuts are taken, and ultimately some people are motivated to commit fraud. In mainland, and I fear it may be true here in Hong Kong, scholars are basically bribed to publish their work in these so-called high-profile journals. Literally, cash rewards are given to scientists for the mere fact of publishing their work in Cell, Nature, and Science. This is a distortion of scholarship. I think it does damage to the scientific enterprise, as reflected, for instance, in the failure of pharmaceutical companies to be able to reproduce key observations in papers published in some of these journals. And let me take a moment to give you an example. Several years ago, the biotech company in California called Amgen reported that most of the discoveries that are reported in these high-profile journals that might serve as the basis for discovery of new cancer chemotherapeutic agents could not be reproduced. They actually did a detailed investigation of the 50 most important papers that they had hoped to rely on, and they said something like 85% of the key observations could not be reproduced. Now, subsequently, the lead author of this study arranged to speak with one of the contributing authors of a paper at a meeting, a cancer meeting, and at breakfast he said to this fellow, "You know, we tried to reproduce your experiment 50 times and it never worked." And the guy responded, "Well, we did the experiment 6 times, it worked once, and that's what fit best into the paper." Now, I mean, this is fraud, and the damage that that does is incalculable. It diminishes the influence that we have in the public perception of science and scholarship, and it harms our ability to encourage the government to continue to invest in basic science. And I submit that at least part of the blame belongs with these journals and their professional editors who are scouting out the seemingly most sensational discoveries. And so I feel very strongly that we should not be held hostage to this process. And one way of doing it is to emphasize publication in journals that are run by active scholars as opposed to professional editors. And I happen to be the editor of one such journal that I've been promoting.
All right, because of time constraint, one more question, one last question.
Considering retirement, what should one consider? Should we try to work till the very end?
That's always a tough, very personal choice. Is there a mandatory retirement age here in Hong Kong?
Yes,
Yeah.
60, and then
60?
you can apply for extension up to 65.
65. Well, I'm 67, and I have no intention of retiring anytime soon. But in the US, we've gone to the other extreme. There's no mandatory retirement, which I think is a bad thing. I think there should be a retirement age, whatever you choose to call that. But people who can continue to do active scholarship and to gain support for that from a government should be given the opportunity after they retire to continue their research program. But I think it's crucial that at a certain age people do retire to create positions for young people to begin their career. I mean, turnover is a really important thing. You know, older folks, people like James Watson who, you know, go off half-cocked, or Linus Pauling, you know, retirement is really important because young people coming up have fresh ideas and they need an opportunity to express themselves. So I think retirement is a good thing, but people should, if they are still competitive, have a chance to continue.
All right, I think we have to stop here. May I invite Professor Jimmy Yu come up to present the question to Professor Randy Schekman.
May I also invite Professor Wong Kam-po, Director of School of Life Sciences, CUHK, to have a group photo.